technically we're afternoon, so good afternoon, everybody. I hope you're having a good Wednesday so far. I'm going to first go over a few things that are due, and then we'll go into the next unit of material. As a reminder, my teaching strategy here is to keep the videos short. So I hopefully won't go over 20 minutes on material, or aim for 20 to 25 minutes of material, and then I've given you a bunch of activities on Canvas as well as readings in the textbook to do to fill your time. The other thing I want to mention again is attendance or attendance points. Attendance, I'm not going to take attendance in the WebEx. Instead, attendance will come from everyone participating in that one discussion board by the end of the unit. So the discussion board has 8 questions that I want you to think about, I want you to consider, and then that will be your discussion, or your attendance grade will be participating in that discussion board by the end. Okay, so with that being said, I'm going to jump to Canvas now. As always, if you have a question, you can put it in the chat feature. Okay, so this is what Canvas looks like for you. If you go to your assignments page, it has the things that are coming up. Keep in mind, I posted that announcement. Please read it about all of the things for lab and the strategy for lab. The lab is a 3-part activity. I broke it up so that there's a part 2 every week. You are more than welcome to get it all done ahead of time or in advance and just do it in one chunk or spread it out however works best for you. Before you do the lab, there's a pre-lab quiz. I posted a video yesterday of myself going through the lab slides, but you can also just look at the lab slides yourself, look at the handout, and take that quiz. And then the first part of the lab activity is due Thursday by midnight. And in the lab handout, it goes through what you need to submit to Canvas. So some stuff to read, there's some data I want you to analyze, and then there are some questions I want you to answer, and that you will submit for the first one. The other activities are what I expect you to be working on for what would amount to the rest of class time. These are things that I would have had you do in class, some of them, and because we're not meeting as a class, you can use your time to do some of these other activities, short homework, a quiz based on the reading from the beginning and the end of chapter 10, and then drawing a picture and submitting some beautiful science art. Okay, I'm gonna switch. Oh, let me just show you the discussion board as well while I'm here. Okay, so Unit 4, questions to discuss. That is the discussion board I'm talking about. That will give you credit for your attendance points here. And it's a little long, so there are 8 questions. And as we go through the unit, you'll be able to answer these questions. So I suggest you keep a running Word document where you just have these questions, and as we get to the material, just write up your responses. For this, I won't take off points if your answers are incorrect. It's more to just get your ideas flowing and get you thinking about these concepts. Okay. So let me attempt to switch gears here. Okay, are there any questions about what is due for lecture activity or lab activities for this week? You guys can put them in the chat, or if you prefer to unmute yourself and ask, you can do that. I clear the tickle in my throat. Okay, I'm going to take that no questions, and we're going to switch to the PowerPoint here. So we had left off, we talked a little bit very briefly about different strategies that animals use to deal, or different considerations animals have for dealing with salt and water balance. And today we're going to dive into that a little bit more. And we're going to talk about nitrogenous wastes and osmoregulation. In your book, this is pages 490 to 495 in chapter 10. And then we'll start talking about the kidney and the nephron. This makes up the bulk of chapter 10, pages 496 to 506. So our objectives for this first part are to compare the excretion strategies in reptiles, birds, insects, fish, and mammals. And then we're going to describe the costs associated with each of these strategies. So the first thing you need to know is that we have waste products. And where do these waste products come from? Well, ammonia is a toxic byproduct that's made from the breakdown of amino acids. And you can either excrete this ammonia as ammonia itself, urea, or you can excrete it as uric acid. So there are three choices for nitrogenous end products that animals use to deal with the ammonia that they make when they break down amino acids. So everyone breaks down amino acids, you make ammonia, and you can either excrete it in ammonia as uric acid or as urea. And we call different organisms, we classify them based on which type of waste they excrete. So if you secrete ammonia, the issue with ammonia is that it's super toxic, so it can't be stored long term. The picture here is of my favorite invertebrate, the nematode. This is what I work on in the lab. And they don't actually store any waste. It just continually flows out in a little secretion stream from their posterior end. And the issue with ammonia is that it also needs to be diluted. So ammonia you can't just release by itself. It has to be diluted in water. And so animals that secrete ammonia, not only can they not store their waste, they also have to use a lot of water to make ammonia able to be excreted. And so this is really only done by animals that live in an aqueous environment. So things like simple invertebrates or aquatic mollusks are going to use ammonia as their waste product. Things that use uric acid, well, on the con for uric acid is that it takes a lot of energy to make uric acid. You're going to take ammonia, you're going to go through some fun chemistry, which I'm not going to talk about because I don't find it that fun, but chemistry magic is going to happen, and you're going to use a lot of energy to get to the state of uric acid. The good thing about uric acid is that it's much easier to store, and it's much easier to secrete without as much water. And so the picture is showing you some bird feces, and you can see the uric acid gives it that nice white glow. And excuse me. And then there are like food bits. Second, I apologize. My spring allergies are out of control. Okay, so uric acid is used as the strategy by terrestrial mollusks, by reptiles, and mainly by birds. We think of birds as using uric acid. This also makes bird pee, poop, bird excretion very acidic, which is why it's damaging to your car. And finally, we use urea. So it's got the same pros and cons as uric acid. It takes a lot of energy to make, but doesn't use as much water. Most animals will predominantly use one of the three strategies, but there can be switches depending on their development. So animals, for example, that start in the water and move to the land might switch their strategy, as well as environmental conditions. So you might change strategies depending on whether you're in suddenly a drier environment or not. Okay, so now what I want to do is I want to start talking a little bit about nephron structure and function. And this is one of the key topics for this unit. And so our objectives are to define the role of the kidney in homeostasis, to be able to draw a nephron and label its major components, to describe the things that occur in the nephron, the processes of filtration, reabsorption, and secretion. And we, again, want to focus on the relationship between structure and function. How does the shape of things, how does the cells, how do the channels and the pumps, how does that allow these key processes to occur in the nephron? And then finally, we want to diagram the flow of solutes in water in each of the regions of the nephron. So what happens in each of the segments? And questions to consider. So how do we rid our bodies of nitrogenous wastes and filter our blood without losing large volumes of water? And how does the structure of the nephron specifically allow for the concentration of this urine? So that's overarching questions that you need to keep in mind as we move through this unit. Okay. Function of the kidneys. So most animals use some sort of tubule to collect and pass fluid that will excrete their waste. And besides just excretion of nitrogenous wastes and toxins, the kidney has a wide array of diverse functions, all under the homeostasis umbrella. So your kidneys are used for ion balance, for maintaining the proper concentration of your different salts, for osmotic balance. This will be involved with the control of the volume of the urine. So the balance of water is related to how much urine volumetrically we excrete. As we'll talk about, there is also an intimate relationship between the kidneys and blood pressure. So controlling volume in urine and the relationship it has to volume in blood will help with blood pressure and controlling blood pressure. We're not going to spend too much time talking about the role of the kidney in pH balance, but it is involved with pH balance. Excretion, this is the obvious function of the kidney, right? It's going to be the way that we get rid of those wastes and toxins and byproducts that we don't need anymore. And finally, the kidney also makes its own hormones that help control all of these processes that we're going to discuss. So let's first start with some gross anatomy. If you look at the anatomy of the mammalian kidney shown here, it's got an outer cortex and an inner medulla, and the urine is going to move through a system of tubes that start small and get bigger. So we go from a minor to a major calyx. Eventually the calyxes come together into a ureter, and the ureter will funnel urine into the bladder. And the goal of the kidney is to take, or one of the goals of the kidney, is to take stuff out of the blood and put it into the urine. And so because of that, the kidney is very vascularized, and there's a lot of blood flow. And in humans, on average, renal blood flow, again, renal just means kidney. So anytime you see renal, you want to think kidney. Renal blood flow in an average adult is about 150 liters of blood a day. So we are processing so much blood in your kidneys. You zoom into the kidney, the functional unit of the kidney is the nephron, and it is made in an adult of about 1 million nephrons. So we're going to focus on the nephron, but keep in mind that it is just a very small part of this organ, and the kidney itself uses all of them to function properly. Okay. So again, showing you that picture zooming in. Keep in mind the gross structure here. If we zoom in, you're going to see all of those tubules with the blood vessels shown in the purple and red. And the functional unit that we're going to spend most of our time looking at is shown down here in the bottom, that nephron unit. So again, the nephron is the functional unit of the kidney. It's composed of a renal tubule, so this is what the filtrate is going to pass through, which is surrounded by a single layer of epithelial cells. So around the tubule, we're talking about the walls made up of one cell layer thick. Now, the cells have different shapes depending on what part of the kidney you're in, and we'll get to that in a little bit. But for the renal tubule, it's one cell layer thick surrounding this open space. As I said before, the nephron, it's very vascularized, and so we've got a lot of blood flow coming in from our afferent arterial, leaving to our afferent arterial, and in the middle where the blood will pass a lot of its waste and products into the filtrate, this occurs at the glomerulus, which is a super twisted structure. It's shown right here. And we have two different types of nephrons that are named for where in the kidney they are. If they are located primarily in the cortex, we call them a cortical nephron. And if they're in the border between the cortex and the medulla, they're a juxtamedullary nephron. So the name will tell you where they are, and then the area that they are located in also determines their shape. So we're going to have a longer loop of Henle if we are in the medulla at all, and a shorter one if we're just dealing with the cortex. Okay. As I just said, microscopic anatomy of the kidney, cortical versus medulla area ones, the cortex, short loop of Henle. These are the most numerous. We're talking 80 to 85% of nephrons are in this region. You also have ones that are in that border region. They have a longer loop, and these are ones we're also going to spend a fair amount of time because they drive this osmotic gradient we're going to talk about. Both of them produce urine. Again, showing you what they look like, the longer versus the shorter. So the tubule of the nephron is, we break it down into different segments. So the area where the blood will meet the tubule, and we will pass materials from the blood into forming this filtrate that we're going to talk about, that occurs at Bowman's capsule. So Bowman's capsule is the area of the tubule that surrounds the glomerulus, which is that tangled up vasculature region. The filtrate will then move to our proximal tubule, then through down the descending loop of Henle, up the ascending loop of Henle, before finally reaching our distal tubule, and then the distal tubule will drain into a collecting duct. Now, multiple nephrons will send their distal tubules into a single collecting duct, which is why it has these little branching off points here. And those collecting ducts will eventually form into the minored calyx, again, minor to major, eventually sending all of that urine into our bladder. Now, again, when we're looking at the nephron, there are two important structural things we want to look at. We want to look at the tubule, where the structures, those epithelial structures that are passing the filtrate along this tube system. But we also want to look at the vasculature. So the vasculature is what delivers the fluid. So blood is going to enter from a renal artery. It's going to come in through an afferent arteriole into the glomerulus. And then the filtered blood, so after it's been filtered, it's going to then move to an efferent arteriole. And the reason that they're both arterioles instead of artery systems going into vein systems is that the arterioles are able to maintain a higher degree of constriction. And so it's easier to regulate the pressure on artery systems than it is on vein systems. The vasculature in the nephron also forms a capillary bed. And so we're going to have, you can see all shown here in purple, we have the vasa recta capillary bed, and we also have peritubular capillaries shown up here on the top region. And so these are going to be where we have that connection point between the filtrate moving through the epithelial tubules, and they're going to send materials back and forth into the bloodstream here in these capillary beds. All of the capillary beds eventually drain into the venous system and will form the renal vein and then go back into circulation. So you need to know, or it's important to keep in mind, not only the tubule structure, but also the vascular structure. So this area meets, or it starts rather, at the juxtaglomerular apparatus. So where the glomerulus is, we have this area that we call the JGA, the juxtaglomerular apparatus, and it's between the tubule and the arteriole. So shown here, you can see in pink, we've got the afferent arteriole with a red arrow showing you the direction of blood flow. So blood is going to flow down that afferent arteriole, A, again for approaching. It's going to come into the glomerulus, and then the filtered blood will then pass through that efferent with an E exiting arteriole. And it comes into close contact here with this region of the epithelial that has macula densa cells. So this is the area of our distal tubule that comes really close to our glomerulus. If we go back here, you see on this picture, the distal tubule is going to come really close to our glomerulus and all of these blood vessels here. Okay, so that area of our distal tubule is lined with these specific cells called macula densa cells that help sense sodium levels. And we'll come back to this when we talk about control, but from an anatomy perspective, I just wanted to introduce you to the different cells we're going to be talking about, because these cells, some of them can secrete things. So in the area between the distal tubule and the afferent arteriole, we have juxtaglomerular cells shown here in teal. And they're going to be able to secrete hormones, which means, which we'll talk about when we talk about regulation of renal function. The other thing that's in this area, this complicated area, are nerves from your sympathetic nervous system. So we've got epithelial tubules here. We got arterioles. We've got Juxtaglomerular cells, we got sympathetic nerves, all in this very, very small region. Okay. We're next going to talk about the processes of urine production. So now that we know kind of the anatomy of the kidney, so what the different parts of the nephron are called, what the different vascular components are, what some of these cells in this small area are called, we're next going to spend our time talking about what happens. What are the functions of these different regions? And there are 4 major processes we're going to discuss. We're going to talk about filtration, reabsorption, secretion, and excretion. And each of these are done in a basic area. So the basic renal exchange processes occur. So filtration is from the glomerulus, blood substances in the blood from the glomerulus going into that Bowman's capsule. Reabsorption is going to occur from the tubules into those capillary beds, into the peritubular capillaries. Secretion goes in reverse. It's things that are passed from the capillaries into the tubules. And excretion is what occurs from the tubules out of the body. And this is showing you visually where those different processes occur. So again, we have that afferent arterial, the glomerulus, the efferent arterial, and filtration are things that are moving from that glomerulus into Bowman's capsule here, forming that filtrate. Then substances that move from the filtrate back into the bloodstream, that is the process of reabsorption. Whereas things that move in the reverse direction from the bloodstream back into the filtrate, that is secretion. And excretion are the things that are moving out. So we're going to use these words, filtration, reabsorption, secretion, and excretion, over and over and over again. So you need to really be comfortable with these definitions, especially the direction that things are moving. With that, I'm going to stop for today. We briefly went over some basic terms about what is occurring in the kidney. We went over the anatomy, the goals of the kidney. What I want you to spend the rest of the time for today is to, again, read your textbook. I'm going to say this every time. You're going to get very sick of hearing it. I think it's really important to then use information and take information from another form. So read your textbook, take notes. I gave you those page numbers at the beginning of the PowerPoint for what you should be working on, and then start working on those activities that are due for this week, okay? I'm going to stop the recording, and then you guys, I'm